I'm very pleased to introduce our guest, Professor Juan Perilla, whom many of you know actually, because Juan was here for many years and was friend with many of you. So by way of introduction, Juan actually did his degree in physics back home in Colombia, and he worked on quark top production and detection actually back then. Then he did a PhD with Tom Wolfe at Johns Hopkins University, during which he works on conformational transitional proteins. And then he joined Claus's group in the TCBG to do a postdoc for several years before joining the faculty at University of Delaware, Department of Chemistry and Biochemistry, from which he just got tenured there from assistant professor to associate professor. And I'm sure he's going to go through the ranks. Well, while he was here with Klaus, he started working on viruses, who you might know actually the cover page of Nature about the HIV capsid that they published together with cryo-EM collaborators. And that's the line of work that he continued to work on over the years. And if I understand correctly, he's one of the few who is working computationally on the subject. So I expect to hear more about viruses today. As the seminar title suggests, Retrovirology through the computational microscope. Thank you very much for coming all the way, and look forward to the seminar.
Thank you.
Please.
All right, thanks for having me. So first of all, that's not a retrovirus. It's not an old virus. So as Imad just mentioned, I've been working on viruses for several years. And for whatever reason, I always find myself going back to retroviruses, and primarily HIV. So this is a figure that Jody, who was also a postdoc in Klaus's group, that's what happens to be my wife, made for a journal cover. And it kind of shows a little bit how we see ourselves, effectively taking dollars and making viruses. Skill. Yeah, building these models from a different sources of information, but always the objective is that we can interrogate and validate these models. Oh. There you go. And as Matt mentioned, we combine a lot of experimental data, including NMR, cryo-electron microscopy, and we're always looking for new ways to learn new things about the virus. Ultimately, our goal is to understand the virus. As many others, we aspire to make these acquisitions into molecular models that we can build, validate, and again, interrogate. So we can learn new things about the biology of the system. And this is inspired, as many others have been inspired here, by the work of Anton Leeuwenhoek, who, rather than pointing to the stars, decided to look at cells. And this is actually what he saw. This is actually his drawings from 1677. So by doing this, he actually started a new field, which is the field of microbiology, right? And this is an attempt, modern attempt to establish what was that he saw back in the day in terms of using these very primitive microscopes. And I'm pretty sure many here have used the supercomputer named after Anton Leeuwenhoek. So we try to use the same principle to try to understand the life cycle of the virus. As Matt mentioned, I started working on capsids more than 10 years ago, 13 years ago, particularly the HIV-1 capsid. At the time, the view on the capsid was a little bit, and by a little bit, I was very critical. Like, people didn't believe it did anything in the cytoplasm. So people thought that we were going after a red herring. And there was a lot of opposition, because the basic view was at the time that the capsid would make it into the cytoplasm and will disassemble immediately, playing absolutely no role. So there was two events that characterize retroviruses. One is this reverse transcription. Reverse transcription is accomplished by reverse transcriptase, RTC, and it consists in a process where the RNA is reverse transcribed into DNA that ultimately forms the pre-integration complex and ends up in the target cell. If you fast forward to 2022, the capsid has become a pivotal point of our understanding of the virus, but we still don't know a lot of things. But there is a lot of potential for drug development, which is what has motivated a lot of the funding agencies to support this work. You can see here they're entry inhibitors. They're not, originally were not thought to be related to the capsid, but now we know the capsid plays a role during entry. Reverse transcription inhibitors. There's also very important and very timely event in the past 2 years. It has been established that the capsid does dock to the nuclear pore complex. So that view that the capsid will disassemble at the beginning has been changed entirely in the past 10 years because there appear 2 new processes, and we have worked on these 2 processes, but I'm not going to be talking about them. One is cytoplasmic trafficking, and the other one is nuclear import. In the past year, there's been a lot of debate whether or not the capsid itself can make it through the nuclear pore complex. And that view changed dramatically in the past 2 years for 2 reasons. And I'm going to show some of that data. One, it was observed experimentally using CLEM. HIV-1 capsids docked to the NPC. And second, it was established recently that the models, and Alex knows this better than I do, that the models of the NPC were too small. So everybody in the field thought that the capsid was too big to go intact to the pore. So today I'm going to be talking about another process that my group is interested in, and we recently got funding for, which is the maturation process. This is the last frontier. There is not a single maturation inhibitor in the market. And I'll be talking a little bit later about the capsid inhibitors and the success of those campaigns and how we can have contributed to that. So my first talk is going to be, obviously, we have not completed the work on maturation. We're planning to do that in the next 5 years. But I'll show you a little bit how our simulations are setting stage to get to that point. So something that people are familiar with here is the idea of doing full-scale dynamics, which is, rather than simulating isolated components, simulating the object at full scale. We've done significant advances by looking at complete assemblies, and we've done significant contributions to the retrovirology field. Back in 2017, with Klaus, together with Klaus, we posited that the central pores of these capsids were able to transport certain ions. Then we proved computationally experimentally, that they could transfer nucleotides. But in this case, I'm going to bring your attention to these collective motions that we observe here. These collective motions, these were global dynamics, blue representing regions of the capsid that were coupled, red also regions of the capsid that were coupled. So this really inspired us to continue working on full scale and try to obtain a better knowledge of the system as a whole. So back in the day, together with Tyler, we set together a goal to build a virion or an HIV. And as everybody who's built a virion knows, this is a very tedious process. And again, this was 2016 technology. So we were able, finally, going through a lot of work and moving from one computer to another, we're able to produce this lipidom, this liposome that include complex dynamics. And that's what I'm going to be talking about right now. So why HIV is good to build these systems is because the lipidomics are extremely well done and has been known for years. So the first time the lipidomic profile was produced for HIV was in 2006 in the lab of Hans-Georg Kreuzlich by Britta Brugger. Then it was reconfirmed in 2013, and then there was another recent acquisition in 2019 that established the exact content of the budded particle. In this case, there are 2 very important details. One is that it's enriched in cholesterol, but more important, it's enriched on a single myelin compared to the plasma membrane. When we built this model, we introduced an assumption that the virus will preserve the asymmetry of the plasma membrane from the body cell. I mean, we really don't have evidence that that's what's going to happen, but that's how we build the model. So our model has different compositions in the inner leaflet and the outer leaflet. We used 24 lipid species to build the model. You can see here in the inner leaflet and the outer leaflet have different compositions. Some lipids are present only in one of the leaflets. And they match stoichiometrically the composition that was derived by Richard Ruger. You can see it's enriched in cholesterol, almost 45% cholesterol. And it also is enriched in single myelin. So to our surprise, the first thing we noted is that lipids from the outer leaflet were moving towards the inner leaflet, and lipids on the inner leaflet were moving to the outer leaflet. So now the timescale of this, when I show this to Rich Pastor or those guys at NIH, they'd say it should take weeks, right? So what's going on? And this is your Leninger view of lipid transfer diffusion. So we were a little skeptical, and this transverse diffusion, our first thought is our system is out of equilibrium. So a detail that I might have omitted is we're equilibrating the system for 3 microseconds. We run the system for 5.2 microseconds. We thought that because the asymmetry in the composition, the flipping was due to that change in chemical potential. But when we look at 0 nanoseconds and 5.2 microseconds, we didn't see a considerable change in the lipid composition of the inner leaflet and the outer leaflet. So even though these translocation events were happening, the overall concentrations were not changing. So the system is in equilibrium, and this process that we were seeing was dynamic equilibrium. One second. So this is the movie of the simulation. I was found it very mesmerizing. And if you pay attention to it, there's a couple of important details. And now I'm going to take your question.
Yeah, were any of the lipids that you saw flipping ones that were supposed to be exclusively in one leaflet, or were they lipids that could be in either leaflet?
The ones that were only in one leaflet, they never made it to the other leaflet. And I'm going to elaborate on that question in a second. Yeah. But if you pay attention, following the idea that your eyes reveal a lot, when you look at this movie, you see the appearances of these little islands, right? And they move on the surface of the liposome. And we start wondering, okay, what's going on with this? What's causing that, right? It's very rich. That's why we like to simulate these complete systems. So here is the translocation. So we wrote a little piece of code. My student Alex, who some of you might have met. We did this analysis actually on a supercomputer to make use of the parallel file system. We track the dependency of the concentration of like the rates, essentially the slopes of these plots. And you can see that the rates themselves are directly proportional to the concentration. So you can see some of them, answering your question, don't flip at all, versus others do flip quite a bit. So there's an asymmetry in the flip rate. As a control, this is cholesterol. Cholesterol is freely diffusing from one leaflet to the other. And again, blue is at the beginning of the simulation, red is at the end of the simulation. 5 microseconds, there's a lot of translocation events, but the overall composition has not changed. This is the number of flip-flop events. Again, this is kind of parsing this data. POPC, for instance, does not translocate to the outer membrane. So there is certainly a dependency on the chemical nature of the lipid type in these translocation events. Okay, so that was very exciting. It was unexpected. Well, we worked with Ilya Leventhal for almost a year to try to characterize whether or not virions that were coming out of cells still had this asymmetry. And we have not been able to prove it whether or not it is asymmetric experimentally. But there is further evidence that a human restriction factor that is a protein that restricts HIV, it means prevents infection, it's a scramblase. So there is a possibility that this asymmetry is still maintained and still has biological role in down the road. Okay, so the second interesting observation then here is this lateral diffusion, right? You can just see it in the movie. So we start to characterize, and because we have the coordinates, and we all in this room brag about having the coordinates, right? So we should be able to tell exactly what's going on, right? So here is the mean square displacements. So there are some hot regions and some cold regions. We can identify and track those cold regions, and we started to analyze them. So the first question, of course, when you build a synthetic system like this one is how much of this is an artifact of how I built it, right? So is this something I put it in there and I never fixed? So in this movie, I'm going to show the slow regions have been colored in red. And in blue, you're going to be seeing, and if you track the blue particles, what you're going to be saying is that regions that were very static and compact disassemble, and other new parts appear by themselves. So you can see this, these blue regions form spontaneously during the simulation. So the process is transient, is not an artifact of how we build the system, and it's something that is in equilibrium. It's happening constantly. They form and then they break. So you can, I'm going to play it again. You will see. There you go. It's a transient process, and I mean, it's very, very interesting dynamics. But now that we have a method to track the velocities, we can actually try to find a molecular explanation to the appearance of those islands in the system.
So this includes the capsid, right?
This one does not have the capsid.
Oh,
this one
just
just the lipid. So what happened was,
okay,
we were going for the virion, and then we were equilibrating the lipids, and we started looking at the trajectories, and they're like, okay, this is interesting. So why keep going when we can't explain what's going on?
Because there is a large region of this that doesn't do the green.
Oh, Alex, this is exaggerating the.
Okay.
Like these are like the very. It's if you pay attention here, there's a continuum of velocities. These are the really marked ends of the velocities. Yeah.
I see.
But there's no capsid here.
I see.
Yeah.
Okay.
Yeah, I've noticed that. So here's what was happening when we started looking at those structures. So first of all, we don't see those islands in the inner leaflet, only in the outer leaflet. That's already very interesting. The slow domains this is what's happening. It's forming these cholesterol crystals. Now remember, this is it's very enriched in cholesterol, it's 45% cholesterol. Form these little crystals that then are stabilized by single myelin, which is also enriched in HIV particles. And eventually that crystal ordering disappears. And that's when the island disappears and forms elsewhere. So this was a very unexpected result together with the flipping. We're trying to put it in the context of maturation. So HIV maturation is a very complex process, and it's still completely, nobody really knows how it works. During maturation, the virus goes from this spherical form. And if you also pay attention to the position of the spikes, they're displayed uniformly over the virus. When it goes into its mature form, when you see the capsid, these spikes actually aggregate in a pole here. And this has been seen by multiple groups in cryo-electron tomography data. Our hypothesis, there's a lot of other details that I'm not going to go into, but there is another key player of this maturation process, PIP2, which actually is removed from the membrane during maturation by the matrix protein, which is this blue protein here. Our hypothesis is that the dynamics of the membrane is changing upon maturation that affects the physical localization of the spikes and turns the virus into this infective particle. So if you remember at the beginning, when we start working at capsid and particularly the contents of the virion, everybody thought that this little guy was very irrelevant to all the events of the virus. Turns out it's also regulating the position of the spikes upon maturation. So this process is a prime target for drug development. All right. So I'm going to talk about another aspect of the virus. So this virus is fascinating in that wherever you look, there are so many unknowns, and it's so related to cell biology that you always end up going down a rabbit hole. So this one we were not so motivated by the infective cycle, but we had a very simple question about the cores. How much force is required to break the core? And what is the mechanical strength of the core? And we characterize that. And if we were looking at this more from a protein engineering perspective rather than pure virological task. So we were inspired by AFM, which is a very well-established technique. In this case, they nano-indent the capsid. You can see here, they displace the capsid, you measure the force. And this has been well reviewed by Woodward in this article. What's important is that you have an elastic region, but if you keep pushing, like we see here, at some point the capsid can just break. And that force is of particular interest to us if we wanted what we wanted to do, which is engineer stronger capsids. So AFM is, as I say, is not a new technique applied to virus capsids. It's been applied for a long time. The typical curves, and we're going to be looking at this, have multiple regions. One is this region, which is the approach, the elastic reversible indentation regime, and the capsid failure. People also look at these capsids, use AFM, kind of an imaging technique, to map the topography of the capsid itself, and that gives them essentially this little topography of the capsid. So we work with a collaborator in Ben Gurion University in Israel, Itai Russo, who is an expert in AFM of HIV particles. And we did a little experiment. So first, he scanned the whole particle 300 piconewtons. They gave him this very nice view of the capsid. So you can see there is also both has its conical shape. But then the idea was to crush it. So he applied 5 nanonewtons. Capsid is indeed crushed. But to our surprise, and everybody's surprise, it went back to its original form. So this itself revealed that the capsid is a very flexible container that is able to sustain very strong forces. This is before and after the compression. The forces that it can sustain are pretty dramatic. 5 nanonewtons, 10 nanonewtons, and 20 nanonewtons. And effectively what Itai is saying is this process here. It's crushing the capsid restores shape, and is revealing that it's a very elastic material. But again, like everybody in this room, we brag that we can actually have all the coordinates and look at this in more detail. So actually, with this shape-based coarse grain and with Dave's and Matt help to accelerate the SMD, we were able to simulate the process and actually start to study what is actually going on with the capsid. So this was one of our first nano-indentations. You can see that after deformation, the capsid quickly recovers. It might look trivial now that you've seen it, but when we started working with this capsid, nobody imagined that if you just look at the equilibrium dynamics, you could not predict the elasticity that we're seeing. So we took it a little bit farther away. So up to this point, we only know that the capsid is capable of restoring its shape, right? So in this case, you have the tip, you nano-indent, and then the capsid recovers. We've seen that experimentally happening very quickly, and in silico also happening very quickly. So we decided to prove the mechanical properties of all the positions of the capsid. So if you look at an icosahedral capsid, the different locations are symmetric. So you might expect to get similar readings as what you would do in different regions, but this is highly asymmetric. So we were anticipating to get different readouts in different regions. If you pay attention to this simulation, we're here poking on the broad end. You can see we're poking right on top. These green are the pentamers. These tan are the hexamers. We get these very nice curves. Here, this is force versus indentation. The scale of the forces matches the experimental values very nicely, but it also reveals a new regime that was not observed experimentally. If you look at the first region, you have this elastic part, but you also have this flat region. And that was also unexpected. When you look at the simulation, right, you see that the capsid is responding as a whole object. It's becoming kind of a pancake, right? So it has an elastic regime, and this second part is called the plastic regime. In the plastic regime, the force response is very different than in the elastic regime. And this is a cross profile of that capsule. So as I said, we decided to probe different regions. So we probed the broader end, we probed the body of the capsid in 2 regions, and also the narrow end. So people have been hypothesizing that the capsid breaks at the narrow end during uncoating. And yeah, all this was done in MD3 using the SBCG, Dave and Matt's contributions to the acceleration. Okay, so here's the readout. So the readout is very interesting. Again, position versus force. The broader end, as I showed before, has an elastic part and a plastic part. I forgot to include a set of slides. Okay. The peak force that we see here is 650 piconewtons. And we can see that the capsid is responding as a whole object when poking that particular part. Here, just below the pentamer on the broader end, the response is very different. It has a very short elastic regime and a very long plastic regime. And the peak force is much lower. So here you can see this is like change in curvature, but this declination is maintained after the inclusion. In here, we have a similar behavior. Is not responding so much as a globular, as a complete object, and we only see a plastic regime. We don't see much of an elastic response to this pushing. And in the narrow end, it's purely elastic. It has no plasticity. So you see it's always a straight line with a very strong peak force. So for the musicians in the room, this is similar to having a string, right? So you have these nodal points, and as you get closer, like if you're in a guitar, as you get closer to the nodal points, it's a little bit stiffer than if you're far from those nodal points. So in retrospect, it makes a lot of sense, the response we're seeing.
So the nucleic acid content of the capsid, does it affect this behavior experimentally versus computationally? I guess you don't have nucleic acid.
We don't have nucleic acid. That's very, very good. That's a valid option. So,
but experimentally they have, right?
Experimentally they do have. But the capsid itself, with its before reverse transcription, it's only like 45% occupied. So it's really, really condensed.
I see.
So there is, and I don't have that data in this slide deck, the stiffness of the capsid during reverse transcription does change. So, so this is as I said, this is a very nice story as establishing the material properties of the capsid. But I wanted to put it in the context of the virology and what could be the biological function of the core elasticity. So back in 2016, I speculated that the core would actually interact with the nuclear pore complex. Again, this NPC that I used at the time was a little bit too small. So we did the best thing you can do with these models that I'm pretty sure everybody builds here on their board. We just made it into a journal cover. But it didn't get past that because, but when I showed this at sort of conferences, everybody was curious about, can it squeeze in? So that was kind of like what was in everybody's head. This is the acquisition 2021, Pilos pathogens. This is NPC, the nuclear pore complex. These are the microtubules, and you can see that HIV capsid nicely documented. So that was paradigm shifting in the field. So there were two camps. The whole field was divided. It doesn't get into the nucleus. It does get into the nucleus. Where does uncoating happen? So this kind of set one camp. There's still one person in the camp. It's hard to, and there's been 3 more papers that show this. And now the competition amongst cryo-T people is to get, everybody wants to have a capsid in an APC. And now there's evidence of capsids inside of the nuclear complex, inside of the nucleus, intact. Okay, so, well, we speculated it. I didn't pursue this much more than that. But now we know about flexibility. So we start to think, okay, is the flexibility going to be a regulating factor of infectivity by getting into the nuclear pore complex? Now the problem is, while the capsid is being transported through the cytoplasm, it is being really dragged on by a lot of molecular motors. It's really being abused as it goes through the cytoplasm. It's being pulled, it's being pushed, right? This could be during those events that flexibility is important. So HIV is a very old field, it's very cryptic, because there's a lot of data, very old data that nobody understands what it means. So this is what we knew. So there's this recent evidence that they must enter the nucleus intact or mostly intact for efficient transcription and infection. There was a mutant, and when we talk to virologists, we have to be very careful what they mean. So there is this E45A that virologists call a hyperstable CA mutant. Now, a structural biology might interpret hyperstability by structural integrity. That's not what they mean. You might interpret it as thermodynamic stability. That's not what they mean. So you have to actually go and look at the assay and what they're measuring, what they call stability. So stability really is how much of the CA lattice they get out of a pellet assay, right? That's what it is. But we know that it's hyperstable, it's very efficient for DNA synthesis, but it cannot make it into the nucleus. And it's been cryptic forever, like, 2002 was reported. And nobody's been able to fully explain what's going on with that mutation. HIV is particularly known because it mutates very fast. It's very good at evading the immune system and drugs. And it does it by making single point mutation. So this is a naturally occurring suppressor, which means that this mutation actually fixes the problems of this guy, R132T. Sadly, I assume that you guys are all known with my structure of my system. You're gonna have to take my word for it, that they're really far from each other, all right? They're not right next to each other. And they're not involved in any known nuclear pore complex CA interactions. So this is a very tough, and it has been puzzling the field for 20 years. Nobody's been able to explain what's going on with that mutation. And the crystal structures look identical. Like it's being crystallized, there's no structural change. Okay, so here's what we learned. So we had a hunch that E45A was going to behave differently than the wild type. And our idea is, okay, maybe it's stiffer, it can't get through the nucleus. And we were very excited when we saw this, right? So we can, from our curve, we can extract the stiffness as the slope of the elastic part. Now, what we get from our stiffness are going to be different in different regions, right? And we were very excited to see that E45A does in fact make the capsid stiffer. So that's it. It's making the capsid stiffer. And we went to conferences telling everybody is capsid stiff. However, the data doesn't fit because the suppressor doesn't recover the original stiffness. So that's not it. And some of our competitors saw us at one of these conferences, and they're still saying that it's elasticity that's regulating entry. But this doesn't recover, the second suppressor doesn't recover elasticity. And that was kind of a wrench to our hypothesis, right? Really killed it. But we kept pursuing this idea. Maybe we should have given up, but we didn't. Because we have very rich curves. And again, because we have the coordinates, so we brag having the coordinates. We all do, right? So we started going again down the rabbit hole of material analysis. Hey, what is this region? When is it going to break? We still haven't answered what is the critical force at which the caps break. So you have the elastic regime, you have this ductile response, which is related to brittleness. And at some point it breaks. So we kept pushing the capsids, right? Elastic, plastic, until we saw failure. Now the problem is if you do it too slow, this plastic regime is extremely long. If you do it fast, you get local fractures relatively quickly. And that's what we wanted to look. Is the capsid more brittle? Which is not necessarily the same as being more rigid or more stiffer. So we've run these five simulations where you can see the indentation, the fracture point, and the force required to break it at that point. And you can see that E45A is indeed more brittle than the wild type capsid, significantly more brittle. This is a stochastic measurement and also depends on the regions. So what about the suppressor mutant? Well, the suppressor mutant did restore brittleness. So it's not the stiffness, it's the brittleness that is related to infectivity. And we call it ductility in a recent conference. And we've seen that there is some experimental evidence that it is, in fact, the mechanism of some drugs is, in fact, associated with making the capsule more ductile by doing what they call fractology, the cryo-T people. So we do see changes in the critical indent, in the brittleness. We don't see a change in the critical force, which makes sense with the lack of change in the stiffness. So now, what is the source of this? We look at the network that comes from the SBCG, and again, I make the mistake assuming everybody knows how that works. But essentially, what we see is that for the longest time, we thought that removing the glutamate, by removing the charge, you were impairing an interaction with the MPC. What we have now learned is that you actually, by introducing an alanine, you strengthen the interaction network of this particular group of amino acids via hydrophilic interactions, about 20 kcal per mole. By introduction of the second site suppressor, is removing this arginine with threonine, you don't necessarily restore the flexibility of this region, but you make another region down below to make it more less stiff. less strong. So that gives us a molecular understanding of how it's happening, the changes in stiffness. Okay, so I'm going to summarize all that we've learned through this process. So stiffness, we were very excited to see a change at the beginning, but the lack of this second suppressor restoring stiffness completely threw a wrench to the stiffness hypothesis. Flexibility and stiffness is not what's regulating nuclear entry. These are late reverse transcription levels. This is virology data from collaborator in Vanderbilt. E45 is competent, incomplete in reverse transcription. That means that it can reverse transcribe the entire genome of the virus. E45 cannot make it into the nucleus, even though it's reverse transcription competent. The second suppressor can make it into the nucleus. The infectivity is restored by the second suppressor. And that correlates very nicely with the brittleness of the capsid. So this is also looking at the percentage of broken capsids, repeating all the experiments. This is brittleness and the ductile character of the capsid was regulating nuclear entry. We also recently discovered in the center 2 new set of mutants, the Q63A, Q67A. They also exhibit altered stiffness. They are competent for reverse transcription, but they are unable to make it into the into the nucleus, and they are not infective. And effectively, they're also brittle. Now, there's another very interesting part of the story, which is there are 2 drugs, one which we actually worked with Klaus, PF74, and another drug that I hope to tell you a little bit more in the next slides, lenacapavir. So PF74 is a drug that was developed by, it's a small molecule that was developed by Pfizer, never got past clinical trials. It does affect stiffness. And lenacapavir is a drug that is currently in the market. It does also affect the stiffness. So why do you think it does to brittleness? Also affects the brittleness of the capsids. So there is compelling evidence that is the ductile character, the brittleness of the capsid that is essential to nuclear entry and essential to infectivity. So in summary, this part of the story is the core is a very elastic material. When I first started working on this 13 years ago, I never anticipated it would be this elastic. It can undergo reversible deformation without structural failure, and the increased brittleness results in loss of infection. Unfortunately, we don't have a model yet of how this actually happens. We just have evidence through simulations, virology assays, and the AFM that all these things correlate, but we don't have cryo-ET that shows exactly what's happening to those particles. We know those particles make it to the nucleus. We know those particles don't make it through the nucleus. We know they tend to be more resident near the nucleus. But there is something going on, but we still don't have an explanation for that. But what we've learned is that the capsid elasticity is fundamental. It's encoded in the sequence, is evolved, is tuned, and is very essential to the whole genus of lentiviruses, where they have must invade the nucleus for successful infection. And also provides a nice explanation how those 2 drugs work, lenacapavir. So this is the last part, but perhaps the most exciting part of the story. So as I showed you guys originally, when we started working on capsid, if you go to the virology conferences or the drug discovery conferences, you face a lot of resistance that anybody believed that the capsid will be a drug target. Everybody said structural proteins are not drug targets, right? This is not a target. You're wasting your time. And there had been several attempts by Boehringer Ingelheim and Pfizer to develop drugs, and they've all failed. But Gilead released last year, in 2022, this little guy, and we've been working with this drug for this, we have been working with the non-medical part of this drug called GSCA1 for several years. And it seems it affects everywhere. It affects nuclear transport, as I've shown, due to changing the ductile character. It affects virus assembly and release. It affects capsid assembly, and it seems that it also affects maturation. So why is this important? Is because this drug, well, it has to be injected, but people only have to take it every 6 months. So people who live with HIV now go from a regime of taking a daily cocktail of drugs, to taking 2 injections every 6 months. So it's transformative in the quality of life of people who live with the virus. And a lot of the drug design that went into this molecule was using several of the models that we developed through the years. And now we're explaining how part of it works. So we're responsible for explaining how it affects nuclear. So in summary, I mean, I've shown you 2 stories. We work on a lot of other stories. A lot of the work is done as part of the very, very big center, the Pittsburgh Center for HIV Protein Interactions. We have a lot of collaborators through the years. Obviously, this is the work of talented grad students and postdocs. You guys want to come visit, yes, feel free to do so. We're right very close to Philly, Baltimore, New York, and Washington, so you can come anytime, very welcome. And I'm happy to take any questions.
Very much, Juan. So time for questions.
You have this drug into the capsid and that affect the mechanical property. How many copy of the drug is required for the
Actually it's very low, and it seems so you only need a few, and it seems it only binds to pentamers. Yeah, so it's very very low concentration.
So in your simulations, how many copies?
We have it saturated.
Oh, yeah.
Yeah. Yeah.
So
Yeah, that address those, people's concern of, say, conventional wisdom. You don't want to target the capsid because the copy number of that protein is so many. You don't want to have that.
So
That's it.
okay, so we work on a lot of, when you look at the infective cycle, like I don't have my percentage note here. Is it still staring? Yeah. There you go. When you go to infective cycle, there's so many steps that it is involved, including nuclear trafficking, and that a lot of these regions, it binds right here actually, in between the FPD and CP, like right here, this is the binding zone. So we always wonder why it had so many surfaces, and why it was so asymmetric. The reality is that a lot of the interfaces that are exposed to other cellular factors depends on curvature. So you have this MPD and CTD that is essentially modulating what partners are binding. So the drug is not really accessible to all the binding sites. So you're probably not drugging all of them, only a few. Because we do know, I mean, when it binds to molecular adapters, motor adapters, FES1 and BIF2, and that's being regulated. That's regulated by curvature. Only certain regions bind to specific motors.
Okay, so I have a question about the asymmetry of the lipids, and because the virus doesn't have ATP. So in order to keep the asymmetry, you need to have ATP, or it's going to dissipate at some point, right? It's gonna become symmetric.
That's what we thought,
Okay.
but, it's really not symmetric because it's not flat. The outer
sure.
leaflet,
Okay, I see.
yeah,
The curvature.
that's our current hypothesis.
I see.
Because it's intrinsically, one has positive curvature, the other one has negative curvature.
So by the way, it's not that you have this slide, you know that they now show that the nuclear pore can breathe.
Can breathe and all that, yeah, but it's full of.
So you don't have to squeeze it through this small version of the thing anymore, but it's still okay. Yeah,
So that's still up for debate because, yeah, it can breathe. They've seen it docked and whatnot. The FG repeat still takes up, right? And this thing is decorated by, like, what I had is decorated by a lot of cellular host factors.
yeah, so
So
but
they-
I guess you were trying to connect your mechanical property of the virus to this squeezing, so I'm saying maybe it doesn't have to necessarily be a squeeze through the core. So I don't know the size of this thing, so I mean, do you have to still squeeze it if you deal with the
It's
large
still-
version of
Still
the
a very
core?
tight fit.
It's very
Very
tight,
tight.
yeah, so this thing is usually,
Okay,
it's a tight fit. I don't think is, at this point, when we saw that the stiffness is not correlated with the phenotype. We don't think it's the squeezing that is affecting, is that maybe it's pulling it and it breaks,
okay, I see.
because that is the ductile behavior that's
Gotcha.
being affected.
I have one question, but I'll just ask one maybe of this interest to everyone. So for the first part, which was really stunning, do you see any spontaneous water transports through the membrane?
That's martini, so those waters are huge.
So, no, oh, you have martini, okay.
it's martini.
Yes. I see. Some of them can
It was still an effort, come on.
But then actually, you also have some lipid defects in your membrane. In the animal.
Oh, that's that we fix those problems.
Yeah, what was the problem?
You mean those
Yeah,
little babies?
yeah, these guys. Yeah.
So this original model was built using, that model was built using,
Yeah,
PACMol.
okay,
And PACMol was very stupid about embedding the lipids in a sphere, so it has poles.
mhm,
And I think those poles turn into those
I
things.
see,
Yeah, so it's a pure artifact.
sure.
Yeah.
So there was something,
Yes, thanks for the talk. So is the assumption, I guess, that the brittle capsids fracture while they're inside of the nuclear pore? Or while they-
God knows, we don't know.
Okay, and have you thought about, or are there thoughts, or do you have thoughts on performing experiments with a capsid that was oriented vertically and the force is being applied to either into the capsid? Because I would expect that's kind of a push the pull through the cap.
I think that's a great suggestion. The reason we just did it like we did is because we can compare to the experiments. Yeah, we have not done that,
Okay,
but we squeeze it, compress it radially internally, and it's something similar to what you did, the expanded isotropically, and also pulling from the inside out.
okay.
So we were another outcome of this work is that we have a very nice parameterized model that we can, AFM parameterized model that we can use to test other mechanical properties. So we did from the in-out, yeah, it's 5 times stronger. It is very flexible, so that was also, once you've seen it, it makes sense. Or we didn't anticipate such large.
I guess my other question was, I'm trying to picture this thing going through the NPC, if you've seen videos like an octopus going through a tiny hole. You don't have to put the whole thing through
Yeah,
at once.
I think-
Could you put like an annulus and constrict it and see if you get 2 bolts, will it
I think
survive?
that's what I think is happening.
It's like,
Yeah, it's like a balloon.
somehow it's like flexible enough to like
Yeah, it's a balloon.
make a hole in it that sort of squeezes through something.
Yeah.
Whenever we see these images, and now for the first time I saw an actual radiation density of an HIV particle through the NPC, right? It's always a conical end which comes first. and not the other way around.
I don't think they have enough data to do that, to prove that. I mean, that's an anecdotal data, because there's the one they wanted to show. There's been ones that are what you would describe, the poor and it's just sitting there. Like, yeah.
Wayne?
Is there any cellular machinery that we know of that pushes or holds through the nuclear pore complex?
Not that I know of.
Energy dependent machine. Okay, I see.
Yeah.
So you're talking about like making holes in the core?
No, is there like a molecular motor response?
Active
for?
push.
Oh, yeah, there is, I mean, you know that,
No, but that's not active. It's
Passive. I don't know, yeah.
It's,
so I hear that Martin had, I didn't see this data, but I hear about it, that they have a capsid going through while the NPC is remodeling. Have you seen that data? No. So there's people who claim that elasticity is not, so this is hot in the field right now. This is the discussion. But there are some people who claim it's not necessary because maybe it's too big.
I see.
The new piece of data that maybe the elasticity that makes elasticity necessary is Martin Beck has an acquisition with an HIV capsid docked to the pore while it's remodeling. So, and we know that it hijacks remodeling.
So by the way, size-wise, they're pretty uniform.
Yes, that's
HIV
surprisingly
yes.
uniform.
Okay, yes. Any other questions?
Zan has a question.
Oh, yeah, sorry, Zan, go ahead. I'm sorry.
Yeah, hi, Juan. Sorry, I was a bit late, but both Alec and I were just at a SACOM meeting where one of the co-workers of Martin Beck was showing some of their results. And I get more the sense that they are, scratching their heads a little bit about what was the radius of the nuclear pore that they should really consider, and that it has changed over time, and that there's a lot more variability and flexibility in the nuclear pore complex and its opening. So they still just kept on showing more or less the same picture where it looks like it got stuck in a nuclear pore, and something more had to happen. But something that nobody really talked that much about was, do you understand the interactions of the genome with the capsid now? Do you can you make sense of that, or is it just random once the DNA is in there?
No. It's actually extremely condensed. Definitely not random.
No,
So,
that I know it's condensed, but are is there certain rich, do they know that certain regions of the like AT rich regions have more contact with the shell than other regions?
So I don't think there are specific contacts between the shell and the RNA. Nobody has ever reported specific interactions between the two, but the nucleic acid is known, once reverse transcription starts, it's known to change entirely the mechanical properties of the capsid. So it's doing something indirectly to the mechanical properties. So the only evidence of that is through AFM, really. There's a lot of talk about condensation and how the DNA as it's being synthesized is also being condensed, and at some point is not condensed anymore, and that's what triggers uncoating. Seems to be highly regulated. And they're trying to keep we've been decoupling uncoating from nuclear entry because both seem to have a change in the mechanical, an effect on mechanical properties. So that's we've kept those two stories separated.
Can I ask one more question? You showed this really interesting result at the end of this drug, maybe what's it doing to the capsid, but have you studied enough of the assembly pathway to know how it affects the assembly? And you could explain to people why it functions so well as it does? So
we've not done assembly. So it seems that it affects assembly. I personally never simulated assembly.
Mhm.
That's
do
good
have-
because Greg Voth is doing that.
Yeah, but we're looking at maturation, so I think the story of maturation is a little bit related to assembly, but I think, and we just got a grant for it. And I think there's a lot of data people have overlooked over the years, so that may be related to assembly at some point.
Mhm.
I don't know, I haven't seen Greg's work on assembly in a while. He changes it every time I see it, so.
Yeah, there's supposed to be a paper coming out in bioarchives. Anyhow, Juan, this is beautiful work. It's really exciting to see how much it is developed.
Thank you, Zach.
Thanks, Sam. So any other questions? Over Zoom? No? Okay, so if not, let's thank Juan more.